Hi, everyone. All of you for taking the time and your busy schedule to come to this symposium. Today, I will be discussing a summer research project, which was understanding the relationship between glutamate consumption and inflammatory gene expression through DNA methylation and demethylation. I want to start by thanking Dr. Perez David and Mr. Hill for being my mentors for this project. One of the platform's amazing opportunity in allowing me to conduct summer research. An estimated 20.7 million adults are limited in their usual activities because of some form of arthritis. This alarming statistic outlines the relevance of the issue of determining ways for arthritic individuals to better manage their symptoms, thus improving their overall daily. Furthermore, one type of arthritis that adds to the 25.7 million adults who are limited in their usual activities is psoriatic arthritis. Psoriatic arthritis is a chronic and inflammatory arthritis that often occurs in relation to psoriasis, which is a skin condition in which skin cells build up to form itchy and dry patches on the skin. In the United States, about 1 million individuals have psoriatic arthritis. Genetics is a study of heritable changes in gene activity due to the direct alteration of a DNA sequence. DNA contains the instruction set for our body. So, for example, it is important in metabolic processes, genetic material to make cells, and plays an important role in regulating our genes. On the other hand, epigenetics is a study of heritable changes in gene activity without changing the nucleotide sequence itself. Epigenetics is a study of how your behaviors and environment can cause changes that affect the way your genes work. For example, your environment and behaviors such as diet and exercise can result in epigenetic changes. DNA methylation is an epigenetic mechanism and is the bonding and removal of methyl groups to regulate gene expression. Gene body methylation facilitates transcription elongation, and this results in increased transcription of a gene. Thus, gene body methylation is positively correlated with gene expression and vice versa. What is meant by gene expression is the rate in which DNA is being transcribed into RNA and then translated into the proteins which govern our bodily functions. Inflammatory cytokines are signaling proteins that help control inflammation in one's body. They allow one's immune system to defend against foreign substances from entering one's body. They have significant involvement in controlling immune and inflammatory responses. By binding to specific receptors on cell surfaces, cytokines initiate diverse signal transduction pathways, ultimately impacting gene expression regulation. However, excess levels of these inflammatory cytokines often lead to complications. One of the most prominent cytokines is TNF alpha. TNF alpha is an inflammatory cytokine that is a major regulator of inflammatory responses and is known to be involved in the pathogenesis of some inflammatory and autoimmune diseases. Glutamic acid is an endogenous amino acid that occurs naturally in numerous food sources. It is commonly utilized as an ingredient in various food processes and culinary preparations in the form of monosodium L-glutamate, also known as MSG, which is an additive. However, various reports have revealed that MSG consumption by human beings is directly linked to human metabolic syndrome, obesity, and arterial hypertension, in addition to various damages in the liver, brain, thyroid, and kidney. So after doing background preliminary research, I came down to two main research questions. My first research question was, how does glutamate consumption affect inflammatory cytokine levels in arthritic and normal individuals? And my second research question was, how does glutamate consumption affect DNA methylation in the gene body of various inflammation genes? An arthritic individual who is glutamate sensitive, as well as their child who was not glutamate sensitive and does not have arthritis, were recruited to participate in this study. IRB approval and participant consent was then obtained, and I then communicated with the Jaymie F Kay lab to discuss the blood draws and processing that I would do there. Blood was collected 3 times over a 2-week period. On our first day of blood collection, a baseline blood draw was taken of both participants. Next, both participants consumed glutamate. Then, 5 days later, a post-glutamate blood draw was taken for both people. This is because changes in DNA methylation can be detected as early as 72 hours. Then a week later, a washout blood draw of both individuals was taken. Separation of plasma and PBMC was performed within 24 hours of blood sample collection. PBMCs contained our individual's DNA, and the plasma contained the inflammatory cytokines. Then quantification of these cytokines were performed at the Mauna Kea lab. The QIAGEN blood mini kit was used to extract genomic DNA from blood samples, and the quick start protocol from this kit was followed. DNA was then sheared to a 6 kb fragment length using a Covaris G tube, and DNA was prepped using a ligation sequencing kit, then sequenced on an R10-4 Minion flow cell. However, I want to preface that this summer, we only sequenced one sample, as we used this as a pilot study to determine our workflow following the sequencing. Adaptive sampling of inflammation genes is necessary to sequence for DNA methylation due to the large size of the human genome. So DNA was then base called using Guppy646 with modified base detection of 5-HMC turned on. We used real-time target enrichment of a panel of 226 inflammatory genes using adaptive sampling against a human reference genome. However, a roadblock came with the adaptive sampling as it is new to our lab. Thus, as I mentioned, the sequencing run served as a pilot in order for us to determine a workflow for the rest of the samples going forward. This summer, we piloted my study and dedicated our time and energy in determining this correct workflow. And going forward, data will be analyzed using the WF human variation workflow provided by Epitome Labs. So going back to my first research question, how does glutamate consumption affect inflammatory cytokine levels? So in order to determine statistical significance for my research, a Wilcoxon sign rank nonparametric test was performed. And this nonparametric test is specifically good for my small sample size, which was a limiting factor in my research. Furthermore, the relative increase of cytokine levels between the initial blood draw and the blood draw that took place after glutamate consumption was determined. In addition, the relative decrease of cytokine levels between the high glutamate blood draw and the washout blood draw was determined for both individuals in my research. Thus, we found a statistically significant difference in the relative cytokine levels between the baseline and high glutamate diet, as well as the high glutamate and washout blood draw for our arthritic individual. And this is what we expected to see. However, we found a statistically significant difference in the relative cytokine levels between the baseline and high glutamate diet, as well as the high glutamate and washout blood draw for our normal individual as well. This was not what we expected to see, as we did not expect to reject our null hypothesis for our normal individual. However, these results suggest that there is a potential genetic component involved with this statistically significant increase in cytokine levels for our normal individual. And here is the relative increase in cytokine levels for our arthritic and normal individual side by side. Here, it is evident that in both people, the relative cytokine levels increase between the initial and post-glutamate blood draw, then decrease between the post-glutamate and washout blood draw. So going back to my second research question, how does glutamate consumption affect DNA methylation in the gene body of various inflammation genes? As I previously mentioned, we were only able to sequence one sample this summer because we used it as a pilot study to determine our workflow going forward. But the important thing is that through this pilot study, we were able to determine a workflow to detect methylation in the gene body of various inflammatory genes. Out of the 226 inflammatory genes that we sequenced for, here are the top 10 genes that had the most methylated base pairs. Hence, we can see that at the top, the gene PTGDR2 had 5.8% of its base pairs methylated. And as I previously mentioned, methylation in the gene body is directly related to gene expression. So PTGDR2 was the gene that was most highly expressed at this initial blood draw from our arthritic individual. PTGDR2 is a prostaglandin D2 receptor that mediates pro-inflammatory chemotaxis that are generated during allergic inflammation. So what I was able to conclude from this summer research is that there was a statistically significant increase in cytokine levels post-glutamate consumption in both arthritic and the non-arthritic individual. And we were able to develop a workflow to detect methylation. So my next steps going forward for the rest of the school year is I will be sequencing the rest of my samples, and I'm really excited to compare the DNA methylation of our arthritic individual who is glutamate sensitive and our non-arthritic individual over the 3 time periods. I learned so many things from being a Kay Fellow this past summer. The first thing is to be proactive and to stay on top of things. My second thing that I learned was that perseverance and determination was necessary to overcome the many roadblocks that I encountered. And research is not a linear process, so staying determined was necessary for me to be successful. And although times got difficult and tedious, enjoying the process of research and learning made the experience very worthwhile. My advice to future K fellows is to not be afraid to ask for help. There are so many great resources here at Iolani that asking for help will only help you and your research. And my next piece of advice is to be organized and write down everything in your lab book. This will definitely come in handy when writing your paper. There are many people that I want to thank for making this research possible. The first is Dr. Perez David, Brennan Yamamoto, and the UH Mauna Kea lab for helping me with my blood draws and allowing me to do my blood processing at their lab. Next, I want to thank Mr. Hill for his endless support and help in every step of this project. This project would not be possible without him and his guidance. Next, I want to thank Dr. Chan for her guidance and faith in me, and I want to thank Mrs. Kobayashi and Dr. Mitchell for their support in me and my project. I also want to thank my participants for their willingness to participate in my study. And lastly, I want to thank John Kay and Iolani School for providing me with this amazing opportunity to conduct summer research. Thank you.